Hello everyone, and thank you for joining us for the sixth and final installment of Top Crop Manager's webinar series. My name is Stephanie Crowley, and I'm the editor of Top Crop Manager. I'm joined today by Albert Tenuta, plant pathologist with the Ontario Ministry of Agriculture, Food and Rural Affairs. Today, during our free 60-minute session, Albert will be talking about soybean cyst nematode, focusing on resistance and new updates. We would like to thank Bayer for sponsoring our session today. Alevo seed treatment fungicide from Bayer is the only product that offers protection against both soybean cyst nematode and sudden death syndrome, two of the most threatening pests to your soybean crop. Soybean cyst nematodes cause root damage, making your crop more vulnerable to root diseases such as sudden death syndrome. With 30% of your yield at risk before you even notice the problem, adding Alevo to your seed simply makes sense. Before we get started, I'd like to remind everyone that this webinar is being recorded and will be made available to all attendees and registrants approximately 24 hours after our live broadcast. This webinar has also been approved for 1 CCA CEU in Integrated Pest Management. This session is scheduled to run for approximately 60 minutes. Albert will speak for about 45 minutes, at which point we will start the Q&A period. If you have any questions throughout the presentation, please type them into the Questions tab found on the right-hand side of your screen. Thank you again for joining us for the final session of our 6-part webinar series. Welcome, Albert.
Well, thank you, Stephanie. I appreciate the opportunity to talk today. So I'd like to start today and just set things up here. I will be talking about soybean cyst nematode, and I'll provide a brief introduction into soybean cyst nematode, its biology. I see that there's a number of people from across Canada, some in Ontario who've probably dealt with soybean cyst nematode, but I see others from the prairies and other areas of Canada that may not have seen or touched soybean cyst nematode in the field. We'll also be talking about management opportunities and how to effectively manage SCN. And then I'd like to, as Stephanie mentioned, talk about some of the challenges we are facing with soybean cyst nematode, and particularly in terms of the resistance management side when it comes to the SCN sources of resistance in that. And as Stephanie said, my name is Albert Tenuta. I'm with the Ontario Ministry of Ag and Food out of Ridgetown, Ontario. And when we talk about soybeans and we talk about soybean diseases, we've concentrated a lot of our time and efforts with many of our colleagues throughout North America in trying to determine what the impact of many of these diseases are. And this is just an example of where we do an annual disease loss survey. In 2015, for example, we looked at the major soybean diseases that affect soybeans across North America, including Ontario and the US. And by far, soybean cyst nematode continues to be the #1 soybean disease or yield limiting factor in soybeans across North America. And annual disease loss estimates are pegged at over $1 billion a year. In Ontario, primarily in Canada and primarily in Ontario, those losses are anywhere from $10 to $35 million per year, depending on the year. If we look at the northern United States, states which are more applicable to most of Canada in that, we look at the top 6 soybean diseases. And these ones, soybean cyst nematode again is #1, and followed by seedling diseases, sudden death syndrome, white mold, and phytophthora root rot. Those are the main 5 diseases that we would be concerned about in Ontario on an annual basis. For other parts of Canada, such as Manitoba, Quebec, PEI, seedling diseases, white mold, and phytophthora root rot would probably be the major disease issues that you would be facing depending on the year and so. When it comes to soybean cyst nematode and identification, that is very difficult, and SCN infestations often go misdiagnosed and are not obvious. There's no symptoms at all in some cases. That is why SCN has often been called the biggest yield robber in soybeans, because it often goes misidentified, and for many years disease losses or injury occurs without being attributed to soybean cyst nematode. Some of the causes, as we see in this picture, which could be attributed to SCN infection or other causes besides SCN, flooded areas. We often get calls from producers in that where you've got wet areas or areas like that have stunted the plants, pH, herbicide failures, particularly where you're dealing with resistant weed species in that. Herbicide injury, the browning, the burning that's associated with those soybean leaves are often another indication or a cause or attributed to these injuries that we would see in this field. Lightning, nutrient deficiencies, often you'll see a potash deficiency, a nitrogen type deficiency, yellowing of those beans. These other diseases, Phytophthora root rot, and others as well, could be attributed to the problems that are occurring in this field. And overall, if you look at that field is the primary issue here is soybean cyst nematodes. So some of the warning signs that you should be looking for SCN are areas of stunted plants, poor crop canopy, filling of those rows, areas of chlorotic growth, that yellowing, necrosis or browning of the leaf tissues. In many cases, when I'm called out to a field with soybean cyst nematode, it's because the grower had poor weed control. So suboptimal weed control is another warning sign to look at. And ultimately, we need to get into the ground and into the soil and dig up those plants and look for the white females, those cysts on the roots. And I'll talk about those in a second. But it's also one of the most common SCN symptoms in many cases is no visible above ground symptoms. It's this lack of visible symptoms which allows SCN to build in many fields without being detected. By the time SCN populations build and visible above ground symptoms, such as what you're seeing in this field, that yellowing, stunting yellow patches that we had in that other field, often by that point, the grower's already seen a 25% or more decrease in yield. That is why it's critical to detect soybean cyst nematode infestations before field populations build. So it is advised that you dig up those plants and check for the cysts on their roots. And I'll explain this again and reemphasize this. Do not, and I repeat, do not pull the plants, since this will leave many of the fine roots behind with soybean cysts on them or knock the cysts off. Soil sampling for SCN is another way to determine if you have SCN and what the population levels are, but I'll discuss that a little bit more. But you want to avoid these above ground symptoms. By that point, you're looking at 25% or more in most cases from a yield loss. And these are the beautiful cysts that you often will see associated with those roots. These lemon-shaped cysts occur on those, and this is the most obvious or the best way of detecting soybean cyst nematode is in that field and seeing those cysts form on the roots. So why is soybean cyst nematode such a great disease-causing organism? Well, if you think about soybean cyst nematode, it's basically a microscopic roundworm that attacks the plants of soybeans and some other host plants, which we'll talk about later. But the typical life cycle of SCN includes eggs, juveniles, and ultimately the adult stage, or that cyst. Let's start in this picture here or this slide with the life cycle with the egg, which contains that juvenile worm. The juvenile worms, the second stage or J2 from that egg hatches, and they're attracted to the actively growing parts of the roots, which eventually they'll penetrate near that root tip. It is once inside that the nematodes establishes a feeding site. It is this feeding site that allows the nematode to feed itself. At this point, it's no longer mobile. It starts to swell and will eventually reach the adult stage as a lemon-shaped female down the road. The male and nematodes will leave the root or exit the root, and he'll fertilize the female nematode at this point and forming that cyst. And what you'll see is that you'll end up getting the eggs being fertilized. There's two different eggs are produced either in a gelatinous mass outside the female's body. It's these eggs outside or in this gelatinous mass that infects the root that same year. The rest of the eggs are kept in the cyst, and they can infect, next year, 2 years, or 10 years down the road. Once the female dies, the cyst becomes brown, and the cyst produces the eggs, protects the eggs from environmental stresses such as, heat, cold, dry, wet. And these cysts are such a wonderful long-term survival structure. This whole cycle too only takes in many cases anywhere from 25 to 30 days. In Southern Ontario, for instance, we might get 3 to 4 generations per year. Eastern Ontario and Quebec, more than likely you're looking at only 3 generations per year. It's this ability of the cyst to be able to survive for such a prolonged period of time, and also quick reproduction and its ability to to go through its life cycle in a month that makes SCN such a successful pathogen, and one that is of major concern. And it's this uniqueness of its life cycle in many cases, particularly when we look at its ability to feed itself. That swollen juvenile nematode sets up, as I mentioned, this feeding site. It starts to produce what we call a syncytium. This is where the nematode ends up feeding itself. It's able to develop into that cyst and then ultimately become fertilized and develop into that adult overwintering structure, that cyst that makes it such a great pathogen. This is pretty unique in many cases, and its ability to transform some of those cells at that feeding site is very unique and makes it such a great pathogen as well. And again, as I said, the soybean cyst nematode is extremely well adapted to survive in soil for a prolonged period of time. In some Ontario research or rotation studies that we conducted in conjunction with Tom Welacky and Terry Anderson with Agriculture and Agri-Food Canada at the Harrow Research Station near Windsor, Ontario, we found that cysts survived for over 10 years under fallow conditions. And when we planted a soybean cyst nematode susceptible variety, one with no resistance genes, the nematode was able to infect those plants. And as expected, we still had high cyst nematode populations in those fields, and we had a significant impact on yield. So again, even after a condition where you're looking at 10 years, 8 to 10 years of a fallow or non-host crop in those fields, these nematodes are well adapted to survive, or these cysts are well adapted for survival. When it comes to SCN, you have to think small. These cysts are extremely very small. Here's a comparison of soybean cyst nematode here on the left here, these fine, small, white, lemon-shaped cysts. These are young females here compared to a typical nodule. And you can see how much larger the nodule is compared to the cyst. What I often do, I call it my zit test, where I'll collect those cysts and put them between my fingernails and squeeze them, and they'll pop just like a zit. So we'll call it that zit test. If it's a sand grain or other pieces of debris in that, you won't get that popping with it. And of course, with the nodules, if you cut them open, you see that red hemoglobin type color to it, then you're looking at an actively growing nodule as well. So remember, when it comes to soybean cyst nematode and detection, it is think small. These are extremely fine, small cysts on those roots, and often occur on those fine roots. And again, those fine roots are very delicate and can be left in the ground as well. As I said, soybean cyst nematode is the #1 soybean pathogen that the soybean pathologists and nematologists have to deal with. And you can see in this field some severe injury. This is a slide provided to me by a good friend of mine, Dr. Greg Tilk at Iowa State University. And this, again, showing the severe damage that can occur. And you can see the spottiness, the patchiness, those yellow areas. You can see the dead areas within that. You can see those typical yellow necrotic areas as well. And then you can see other areas of the field that are still green. Doesn't mean that soybean cyst nematode isn't present there. If we dug up those roots, more than likely we will see cysts on the roots, just that for one reason or another, the nematode populations in that might be low, or the plants are able to compensate for that injury on the roots themselves. But again, you can see the high population zones here and the impact. And also, if you notice along the edge of the fields here, those anywhere by which soil can move into that field, along fence rows, compacted areas, stressed areas, all those areas are often areas where you'll see soybean cyst nematode probably showing up first in those fields as well. It's this absence of symptoms, which is one of the big problems with soybean cyst nematode, both in Canada and the US Midwest. In many cases, this is what SCN infestation looks like. If you look at it, it looks like a normal field. You see beautiful, lush green plants in this field. You wouldn't even think that there was an issue here. But in this field, you might be looking at a 5-bushel yield difference between a susceptible and a resistant variety. Again, this is why soybean cyst nematode is often referred to as that silent yield robber. And so what have we seen, or when was soybean cyst nematode introduced in the spread throughout North America? It was first introduced in 1957 and then slowly progressed down the southeastern United States, the Gulf states, and up through the Mississippi. Basically, anywhere soybeans are produced in North America, particularly the US and into Canada now, you're starting to see soybean cyst nematode move in. And you can see quite extensively how it's moved and become a significant issue in terms of its spread across. In Canada, Ontario, 1988 in King County near Chatham, Ontario, actually just a few miles from where I'm based here at Ridgetown, was the first time we saw soybean cyst nematode detected it in Canada. And since then, it's moved from southern Ontario here to the primary soybean producing parts of the province, into central Ontario, Northumberland County, etc., and then into eastern Ontario near Ottawa and that. And then most recently, the first find outside of Ontario occurred in 2013 in St. Anisette, Quebec, just on the other side of the Ontario border, which would be expected. Again, remember, soybean cyst nematode can move by any method by which soil can move. So in this case, maybe equipment or wind blowing of soil, etc., or movement across the water into Quebec as well. So again, soybean cyst nematode has now been detected for the first time outside of Ontario into Quebec as well, and continues to become, spread in that province, and unfortunately will as well. So if we look at the map in terms of distribution in the US and Canada, two areas to focus in on, or where we see the new frontier when we talk about soybean cyst nematode, is eastern, the northeast United States into New York State, and eastern Ontario and Quebec. And then the Great Plains, say the North Dakotas, Minnesotas, those areas there can have a significant impact potentially on other parts of Canada, particularly Manitoba and the prairies as well. New York State, in 2016, a fall sample was collected and confirmed in 2017 to be positive soybean cyst nematode. It's interesting, the same county, Cayuga County in New York State, was also the first one where sun-death syndrome was detected. So soybean cyst nematode and sun-death syndrome are two diseases that often coexist. And if you find one, you often will find the other as well. In terms of expansion or potential expansion of soybean cyst nematode into other parts of Canada, particularly the prairies and Manitoba, as I mentioned, that other frontier or other area that we're seeing expansion is into the Dakotas and Minnesota. Here's a map provided to me by a friend of mine, Sam Markell at North Dakota State University. And you can see that soybean cyst nematode is increasing in North Dakota, as well has started to be found and detected right by the Manitoba border. And so as cyst nematode develops in North Dakota, that increases the risk of the pathogen or the disease, the nematode, coming up into Manitoba as well. Remember, I mentioned the soybean cyst nematode can move any method by which you can move soil: wind, water, birds, equipment, etc. So, the Red River or Red River Basin poses a risk in this case, since flooded areas of the Red River could act as a means for these cysts to move into Manitoba. And so my brother, who's based out of the University of Manitoba, has been surveying Manitoba over the past 4 or 5 years. And as of right now, he hasn't found soybean cyst nematode, or there hasn't been a positive detection for soybean cyst nematode in Manitoba. Of the 487 samples that they've looked at, or his labs looked at, 69 of those turned out to be what we would call nematode cysts. But again, most were damaged, most were empty. Those that were intact, they did find out that they weren't soybean cyst nematode. So, few had eggs and juveniles for molecular analysis. So again, the sample size is there, but again, getting viable plump cysts with a lot of eggs and juveniles to do some of this testing has been difficult. And so those ones that they have looked at are negative. But again, remember, as Manitoba continues to increase in their acres, we're looking at 2 million plus acres, relatively new to soybean production there. But I would expect, and so would my brother, that over the next 5 to 10 years, that soybean cyst nematode will likely be established in Manitoba. Remember, wherever soybean production occurs, soybean cyst nematode seems to find its way there. And so, the severity of soybean cyst nematode depends on a few factors, the pathogen or the nematode in this particular case, weather conditions, and soybean varieties. Us pathologists, we like to wrap these things up into what we call 3 factors, the disease triangle in this case, with our students here, and that we usually call that plant pathology 101. The amount of disease that occurs depends on, how aggressive is that soybean cyst nematode? How susceptible or vulnerable is that variety that we're growing? Is it a more tolerant variety, resistant variety, or is it a susceptible variety? And of course, weather conditions and environment play a critical role in disease as well. So, this whole disease triangle concept works really well to explain yield loss for soybean cyst nematode. The conditions, if we were out in, if I was presenting this in person with you, I'd be out there right now asking the question, what conditions favor soybean cyst nematode reproduction and yield loss? And hopefully you would answer, hot, dry conditions are the key here. So soybean cyst nematode reproduction, can occur very high, and yield loss is extreme, particularly in those hot, dry conditions. Again, stress conditions on those soybean plants can make the nematode impact and injury much more pronounced. And in many cases, that stress can occur even when nematode populations are lower. So stress is an important piece of the puzzle when it comes to determining or the risk or yield loss potential for soybean cyst nematode. So how do we go about managing SCN? How do we do the integrated management for it? First of all, scout for early detection. As I said, the earlier we can determine or find if soybean cyst nematode is present at the lowest population levels, it's easier to manage. So areas such as Quebec, Manitoba, when it's found, if those levels are low, it'll be much easier to manage than when we first detected soybean cyst nematode in southwestern Ontario, when populations were 20, 30, 40, 50,000 eggs or more, which made it very difficult, and the yield loss was quite significant. So if you look at this field here, it looks really good. This, it's in many cases, it's very difficult. You go into a field like this, if you're an agronomist or even a producer going in that field, you're going to think that everything's honky-dory. You're not really thinking any issues there. But again, this field here could have some soybean cyst nematode, so you need to be scouting these fields as well, whether you see visual symptoms or not. Dig up those plants, look at the plants, and look for roots. So whether it's that nice field that we see on the left here, or that severely damaged field on the right, both of these, the left and right, soybean cyst nematode could be churning around in those roots, in the ground there, just building up. So higher numbers are building up. As the numbers build up, the greater yield loss potential as well with those. So keep that in mind. So regardless of what the plants look like above ground, their soybean cyst nematode could be doing its thing and song and dance on the roots as well, building up and merrily going about doing its business. So there's 2 ways to effectively scout for soybean cyst nematode. Neither one of them involves driving in this air-conditioned pickup, which is very similar to mine. So again, you can't do it at 100 kilometers or 60 miles an hour. You need to be out there, scout those fields, and whether or not, as I said, whether plants look sick or not, you need to be out there sampling and scouting for soybean cyst nematode. And two ways of doing that is dig those roots, look for the females. Don't pull them because there's issues associated with that, or collect soil samples for testing in the lab as well. And so digging up those plants, what you want to do is be very careful. Again, use that shovel, never pull those plants up. dig those roots up, shake the soil off, and look for the white females on those roots as well. As I said, if you pull those plants out, you end up leaving a lot of these fine roots where soybean cyst nematode is detected in the ground, and you may get a false negative out of there. Again, this picture here shows, slide shows again that relative size between the nematodes, these white pearl nematodes or cysts on the roots, compared to the nodules on there. So, digging up the roots, looking for the females are, the advantage here, of course, is it's quick, it's cheap, and it can be done anytime after the nematodes have gone through their first cycle. So generally, usually, I would start digging about 6 weeks after planting. That will give the first generation to develop from cysts in the soil to cysts on those roots as well. And in many cases, you can do that until, into August or so, or just before harvest, one thing you may notice is that the nematode numbers or the cysts on the roots may decline when the temperatures are 90 degrees Fahrenheit or higher. That could lower the slow down the reproduction on those as well. So the other thing is, don't scout once. Don't dig up those plants once. Come back, say, a couple of weeks after, and you may find that there are more nematodes on those roots again, because you might be hitting the cycle in between the development there. So don't scout once. That's called inspection. Scout more often. Get out there 2 or 3 times and look at it. The only problem with this method is that it doesn't give you a quantitative number. It doesn't tell you what the numbers of cysts are in that field, but if you're trying to detect whether or not cyst nematode is present or not, this will do the job and that. And so how do you go about soil sampling to get more of a quantitative amount? So here's an example of an 80-acre field or so. Divide that into 4 20-acre cores or acres or blocks in there, and you can take when you do a fertility sample, that's a great time to also do a soybean cyst nematode. As you do your zigzag pattern through the field, taking samples, in this case anywhere from 15 to 20 cores at 20 centimeters or 8-inch depth is ideal. You can split that sample for a fertility sample. You can also send that off to one of the soil testing labs for soybean cyst nematode as well. So that's one way. Just break them off into quarters. A simple, easy way to do it. You can also, if your system is based on say management zones, or your field's irregular or so, you may want to go into break it up differently into different areas depending on these areas. So you may want to, in this case, break it up into 3 or 4 different areas as well. They're just as acceptable as doing the rectangle earlier, or ideally, you can also try to, if you're specifically really looking for soybean cyst nematode detection or so, this also is another neat way of doing it, is just go to those high-risk areas. These high-risk areas include field entryways, where soil falls off implements or equipment that's moving into that field, particularly those entryways and that. High pH areas, soybean cyst nematode in some cases like areas where there's higher pH in those areas. Those low areas of the fields or flooded areas where, again, water movement could move cysts into those areas as well. And also those lower areas might have higher populations because you're moving more cysts in there. And one area that we often will also see is near buildings, equipment, storage areas. Again, that soil will fall off, very similar to what you'd see with field entryways, and then along fence rows. Again, particularly in the direction where the wind blows into those fields, soil, snow, etc., will blow those cysts into the field. So these are some of the hot spots or risk areas to concentrate on as well. It doesn't matter how you go about sampling. The important part is sample. Find out what those levels are with soybean cyst nematode. It'll give you an indication of what the pressure is in that field, but also it will give you an indication later on as you put your management practices to use, say with resistant varieties, etc. It'll give you a way to evaluate whether your nematode numbers are going up or going down. It'll give you an idea of the effectiveness of your management program. And so also in terms of when to sample, that's a question we often get. You can do it in the spring before you plant soybeans, in the fall, particularly after you've had a soybean crop in there, and for maybe you had some yield issues, or you saw some low areas or some impacted areas during harvest or on the combine. So you may want to get out there and sample. If you're planning to grow soybeans next year, that fall sampling when you have, say, corn or wheat or a non-host crop in that field is, again, another good reason or a great time to sample that field. And as well, if you're just trying to find out if you have soybean cyst nematode, you can do that anytime during the season to find out if cyst nematode is present or not. And why is it important? As I said, it's a means to evaluate your management, but it gives you an indication. The higher the cyst nematode levels, the cysts in the soil, in this particular case, we're looking at cysts on the roots, but as you get higher populations in the soil or the cysts on the roots, there's a drastic decrease in yield over time. So again, soil sampling can help us. Knowing your numbers can help you tell you whether you're at a higher risk or lower risk, or what the impact potential is in that field as well. Non-host crops is another effective cornerstone or part of the management of soybean cyst nematode. Non-host crops, and particularly for say Ontario, corn and wheat is an effective non-host rotation in there. What you want to do is avoid growing continuous soybeans or including soybeans and dry beans together, because both of those can be hosts. So the more time soybeans are in the rotation, the greater potential for SCN levels to build up in the soil and ultimately decrease yields as well. So remember, those juvenile nematodes or worms, they'll hatch from those SCN eggs, and those hatched juveniles are the weak link in the life cycle. We want the nematodes to get out of that protective cyst and get into this nematode or worm shape here. We want them there because they won't survive without either a food source, and they won't survive in harsh environmental conditions such as the winter, which we continue to seem to have down here, even at this point as April 10th. So, what we've seen in Ontario and the U.S. Midwest states, such as Iowa and other areas, is that 1 year of corn can drop soybean cyst nematode egg numbers down anywhere from, say, 5 to 50%. And that's a good thing. And you may be asking yourself or saying to yourself, I'll grow just 2 or 3 years of corn and really knock it back. Unfortunately, that's not quite the way it works. It's not a true linear relationship like that. That second year corn is not as effective as that first year corn at reducing those SCN eggs numbers. That third year corn is even less effective, or third year non-host crop is less effective. One thing we've seen in Ontario is that wheat seems to be a better non-host crop than corn. So having that corn, soybean, wheat rotation is a definite nice management tool in there. I remember a field near Chatham, Ontario, over like 20 years ago, where a grower grew continuous corn for 8 years because his soybean yields had decreased over 50%. He did not know why at that point until we came in and saw the soybean cyst nematode on the roots. So in year 9, he grew a non-SCN soybean variety. At that time, he didn't know about soybean cyst nematode. We had very limited varieties, and it was severely damaged even after 8 years of continuous corn. When we tested that field, those SCN populations in that field were still over 88,000 eggs per 100 grams of soil. We get concerned at 1,000 eggs per 100 grams of soil. 5,000 to 10,000 is considered moderately to high nematode population. So 88,000 after 8 years of continuous corn, those numbers did not decrease. So you can't base your rotation on just non-host crops in there. So you need to incorporate soybeans into that system, and in this particular case, resistant varieties fall in there. So what are some of the other hosts for soybean cyst nematode, particularly for us? Soybeans, dry beans, there is discussion about many of common weeds out there as being potential hosts. I've looked at in the greenhouse, they show up under Ontario conditions. I've looked at weeds in the field. I've yet to see cysts on those roots. Some other non-host crops besides corn, wheat, sugar beets, forages, canola, barley, all of those are effective non-host crops as well, and also some of the vegetable hort crops as well. I mentioned dry beans. Dry beans are another host. In many cases, maybe the visual symptoms aren't as visual on dry beans. It may need higher populations in some cases for the visual symptoms to show up. Here's a slide from Berlin Nelson at a North Dakota State, just showing how various levels of soybean cyst nematode can definitely impact those visible growth of the dry beans in that as well. So remember that if you're growing dry beans, soybean cyst nematode can infect them as well and cause significant impact. So when it comes to management for soybean cyst nematode, I've talked about early detection, non-host crops. Resistant varieties, though, growing resistant varieties, that's a cornerstone of any integrated management system, and that goes for other diseases as well. Here we're looking at two fields in Ontario, one with high cyst populations over 5,000 eggs. You can see the susceptible varieties on the left compared to two different sources of resistance, Peking and PI 88788. On the right, you can see 50% or greater yield loss and considerable amount of symptoms in that field. Another field, this one down in Essex County near Windsor, where that susceptible variety, lower cyst populations, 10 bushel loss or roughly 20%, but less visual symptoms, very little visible symptoms again. In this particular field, again, you may not have even known that it had soybean cyst nematode, but you're losing 10 bushels or more in that particular case. I mentioned that study that we did in conjunction with Tom Welacky and Terry Anderson, where we took growers that had various from low to high soybean cyst nematode levels in their field, and we incorporated resistant varieties in green triangles and susceptible varieties in those rotations and got them to incorporate non-host crops, so corn and wheat in the rotation. And over time, what we found is that we slowly started moving our cyst nematode numbers down. You started seeing, particularly with the resistant varieties, those green triangles, we started shifting farther and farther over to lowering our populations. And eventually, the best combination, of course, was using those non-host crops, those corn and wheat in that rotation with the resistant variety, was the most effective means by which we could lower our cyst populations and bring back most of that yield back. And those areas where we had that susceptible variety, again, we're still the non-host crop rotation was lowering them, but not as quickly as if we incorporated those resistant varieties into it. So what are some of the challenges we're facing right now? SCN continues to spread. Its aggressiveness or virulence is also increasing, and resistance is failing in some areas. And part of that is we have too few sources of resistance, and also growers have been managing soybean cyst nematode in Ontario and the US, so maybe that grower awareness and concern isn't the same as it was 20 years ago or 25 years ago. Most provinces in Ontario and states in the US do performance trials just like we do in Ontario. If you look at this year's soybean cyst nematode varieties out there, you'll see that in Ontario, again, if you look at our sources of resistance, there's the 88788 source of resistance or Peking. The vast majority, though, are that 88788. These are looking at our Roundup Ready varieties. If you look at the yields on these varieties here, those Pekings, they're high, they're effective. This again is on our compared to our susceptible checks, which would be about 45 bushels or so down here. All our resistant varieties would much more provide a better yield or manage soybean cyst nematode better than the susceptible checks. These fields would have been 3,000 to 6,000 eggs per 100 grams of soil. We're starting to see some of those cyst populations that can reproduce on these other varieties here that have the 8.8, 7.88 source of resistance, and you can start seeing the Peking starting to come up. When it comes to conventional soybean varieties, we don't even have that peaking source of resistance in here. We're dependent solely on the PI 88788 source of resistance. And this, again, can put significant pressure on these varieties and that 88788 source of resistance. So, when we look at what we have available out there, although we have many different sources of resistant sources out here, PI lines, the Peking, the Hartwig, the 88788, and those, we are still very much dependent on this 88788. It's 95% of all soybean varieties, whether you're grown in Ontario, the US and that, they're very dependent on this. So this reliance on the 88788 has put enormous selection pressure in favor of SCN. In general, as I said, those 95% of our varieties that are available to our producers out there are dependent on that one source of resistance out there. Although there are many other sources out there, plant breeders have had probably over 100 different breeding lines with different sources of resistance to SCN have been identified and are available to breeders. And these lines can be very effective against this nematode. Unfortunately, when it comes to agronomic desirability, they are dogs in many cases. They don't yield very much. They have flat viney growth to them. They're black seed coated. They're late maturing. All those things that we want from agronomically accepted soybean varieties in terms of having good yield potential, good maturity, cyst nematode resistance, etc. These lines or breeding lines have not been able to cut the mustard and being incorporated in there. So although there are many of these breeding lines, we're still basically dependent on that Peking and most definitely that 88788 source out there as well. And when it comes to soybean cyst nematode resistance, what we're talking about in this case is the amount of nematodes or female nematodes, the cysts on those roots, basically the reproductive ability of that resistance to minimize that female reproduction on itself. So if we look at compare a susceptible variety and say we have 100 cysts on those roots, and we compare it to other varieties, if a variety has 10 cysts or less on those roots compared to that susceptible check, we would call that a resistant variety. And so the varieties that are out there by definition basically mean that they have 10% less reproduction on those roots compared to that susceptible check. And so again, that is part of the problem here, is that SCN is adapting to that 88788 source of resistance. So we're starting to creep up above that 10% on there. And this adaptation, you can see just by the numbers here, is increasing in natural SCN populations across not only Ontario, but particularly the mid-central United States. And this map shows those results of surveys conducted in numerous states in the US as well as Ontario of the level of reproduction on SCN populations, with the pure 88788 source of resistance. And so you can see Ontario is relatively low at 33%, but still a very concern there. Other areas that have had a lot of cyst nematode, a long history of cyst nematode, the Missouris, the Illinois, Indiana's, Ohio's, Tennessee's, you can see those numbers are very high in here. So a lot of these fields have populations that can reproduce on that 88788. And of course, this adaptation is a great concern to us all. In Ontario, we look at the reproduction, or that female index on those 288,788. This is work done with in conjunction with Tom Welacky. He's been doing he's a great colleague and been doing a lot of work on soybean cyst nematode over the past 25, 30 years here. And if you look at the resistant lines out there, the majority would fall into that resistance with the 88788 of being below that 10%. We're starting to creep up above that 10 to 30% female index. Again, it's at 22%, but overall for Ontario populations, you know, that 88788 source of resistance is still effective and providing us with an effective management tool in the province. If we look at the average over time, sure, we are starting to see those numbers creep up. We're starting to see reproduction more on those 88788 source of resistance above that 10% threshold. If we look at the other main source, that Peking source, the numbers are below that 10%, and that's where we want to be as well. If we look at Iowa, and a good friend of mine, as I said, Dr. Greg Tilka out of Iowa, has been looking at the variety trial performance locations over the past 25 years. And if you look in the early 2000s, for instance, the amount of reproduction, that percentage was you know, anywhere from 10 to 30%. Very similar to where we are in Ontario right now. What's happened over the past 10 or so years? Those numbers have now creeped up to 30 to 60%. So the number of reproduction, or the number of cysts on that 88788 source of resistance in these variety trials continue to increase. And so now you've gone from being below 10% to below 30%, to now you're looking at 30 to 60%. So that effectiveness is starting to decrease in these fields in Iowa for the 88788. The Peking source of resistance, very similar to what we saw in Ontario. Most of it is below 10%, but again, you're starting to see some greater reproduction above that as well. But again, the Peking is an effective management tool in this case. So does this all look familiar? Of course it does. It's what we're seeing. We had a breakfast meeting this morning where the hot topic, of course, was glyphosate resistance, herbicide resistance, weeds out there. And this is the same thing that's happening right now when it comes to 88788 SCN resistance out there. So what's the prospects for resistance in the future? Unfortunately, you know, it takes a long time, and as I mentioned, having other non-PI 88788 sources of resistance in the near future is going to be difficult right now. So that usefulness of the tradition, you know, these 88788 resistance varieties will probably continue to decline. We'll start seeing more cysts on them unless we do something about it. And that as well. And so how do we go about prolonging or slowing down or keeping that effectiveness of those 88788 resistant sources? We need to rotate those known house crops into that rotation, as I mentioned earlier, as well as rotate those SCN resistant varieties in there. So don't overuse that same resistant variety. Do not use the same resistant variety regardless of SCN resistant source. So if you're find you have soybean cyst nematode, or you're in your rotation and you've gone through, you know, a non-host crop, you're coming in, follow that up with say after your corn, you're going to come in with soybeans, plant an 88788, for instance. Years 3 or years 4, you're going to have a non-host crop in there. It could be corn, if you're going to follow that up with soybeans, or if it's going to be corn and wheat in there. And then following that, you're going to want to have a different SCN variety. Even if it's an 88788 source of resistance, you have to grow the different variety. Or if in this case, you could bring in a Peking source of resistance. And you follow that through again with your non-host, your different SCN varieties, 88788 source of resistance or Peking. And you want to keep that. That's basically to prevent these new SCN types from developing as well. Another common thing that we're getting a lot of attention is on cover crops. And we know the benefits of cover crops associated with soil health, soil tilth, etc. But it's important also to investigate what's the impact on these cover crops could have potentially on pests. This is a study that I did a couple of years ago where we looked at 26 different cover crop species. The good news is that very few are reproduced on or had soybean cyst nematode reproduction on the roots or in the roots as we looked at the roots there. The hairy vetches, some of the clovers, and that had some soybean cyst nematode associated with those roots. But of the other 3 nematodes that we looked at, spiral stunt or root lesion nematode, the Pratolinchus, it's the sun hemp and these, you can see those blue bars have increased considerably in some cases, whether peas, the fava beans, all of these legumes. But the one particular one was the sun hemp, which has been promoted for reduction or management of soybean cyst nematode. And it might be an effective cover crop for reducing cyst nematode. But unfortunately, looking at this preliminary data here, it may be increasing another nematode in the root lesion. And that could increase your risk for nematode injury, not only in soybeans, but corn, wheat, horde crops, etc. So we may have gone from a cover crop maybe that could be effective in managing one nematode, but unfortunately could increase another one now that can affect your crops in multiple years or have a greater impact as well. So again, we need to investigate, and we'll be repeating this experiment, but at the same time, we need to be evaluating these cover crops and their potential impacts on whether nematodes, root diseases, etc. as well. Seed treatments, another tool that we've started seeing a new exciting area of SCN management recently is these seed treatment nematicides. In Canada, we have Clariva from Syngenta, we have Votivo, Olivo, as Stephanie had mentioned earlier, for soybean cyst nematode, and that as well. Nemastrike is a new one that just got registered through Monsanto, and with the diversification, I believe it will be going to BASF as well. So and then there's many other products that are in development. You're going to see that many more products and many more coming from many other companies in the next while in both Canada as well as the US, where they've got a few other products out there for nematode management as well. But again, as I said, we're in the process now. We're evaluating a number of different nematode products out there, and one of the key benefits of some of these seed treatment nematicide products is can they help us and slow down the effectiveness or the loss of effectiveness of the 88788? Can we slow down the buildup of these adaptive populations? Also, the yield and SCN effects of these new seed treatments, in terms of their yield impacts, maybe their impact on cyst nematode, where it's still early in the research side and development side of these. Remember, these seed treatment nematicides are another tool in the toolbox that can help us, but they are not a silver bullet, and that's something to remember and keep in mind. So for SCN management, it's that integrated approach which is most important and critical to its management. And so if we look at integrated management SCN, as I said, collecting those soil samples, know your numbers to help you in terms of your evaluation, your risk factors to cyst nematode. Growing non-host crops, corn, wheat in that rotation will help us in terms of populations. That these new seed treatment options nematicide options are also another tool for that integrated management of SCN. And then of course the cornerstone, those resistant varieties, whether it's the 88788 source of resistance, other sources of resistance such as Peking in that. And then we're trying to also cover crops. That could be a useful management tool in here as well. And we're still, as I said, trying to determine the usefulness and consistency of the results when it comes to cover crops and that. So there are many challenges, as I've mentioned today, in terms of cyst nematode, from the evolving adaptation of the 88788, the nematode, to it, the effectiveness of that source of resistance. It's important to remember that not all varieties are created equal. We are seeing new tools out there. We want growers to actively manage cyst nematode, be proactive in that, and then ultimately we need to work with industry and growers and the whole soybean stakeholders, all stakeholders, to manage cyst nematode and have one voice, one message to meet these challenges. And that's where we've launched this new Soybean Cyst Nematode Coalition, and you'll see a lot of this. Take the test, beat the pest, SCN Coalition materials and that over the next year or two, and again, we're trying to promote that one voice, one message, a unified approach to meeting this SCN resistance challenge that we have right now. This the coalition was launched in February at the Commodity Classic this year, and was quite successful there, and we've got a number of resources. We've got a website, the Soybean Cyst Nematode Coalition.com resource center. There's a number of different materials available, resources available to help both agronomists, growers, and others in terms of managing soybean cyst nematode and that. And you'll see a lot more presentations, informations, videos, etc., both from the SCN Coalition as well as from people like myself, other provincial pathologists and state pathologists as well. And again, we're trying to build partnerships, and not only with the soybean checkoff groups such as the United Soybean or the North Central Soybean Research Program, also corporate sponsors there as well, but also looking at local state checkoff boards too, such as the Grain Farmers of Ontario, and many of the boards in the local state boards in the U.S. as well. Again, coming on board and supporting this effort. And basically, what we're doing is just re-emphasizing the best management practices when it comes to cyst nematodes. Growers working with their advisors to develop a plan to manage soybean cyst nematodes. Know those numbers, know where your risk factors are for soybean cyst nematode by starting by having an evaluation of what's going on in those fields, and also gives you an opportunity to evaluate your management. Rotate those resistant varieties, rotate those sources of resistance, rotate those non-host crops such as corn or wheat in the rotation, and then also start looking at these seed treatment nematicides as another tool in the toolbox to help us in terms of our best management practices as well. We've had I've talked a lot today. It's not I've got a lot of colleagues and grower groups to help fund or to help with the work here. It's a great group in terms of both in Canada and the U.S. in terms of coming together to bring us the best information to help us in terms of managing the challenges when it comes to soybean cyst nematode as well. If there's any questions, I'd love to answer some. Thank you.
Thanks so much, Albert. I do have a few questions here for you. So to start out here, are there certain soil types that soybean cyst nematode like?
Yeah, so that's a question we often will get, and in many cases, soybean cyst nematode will reproduce and build up in any soil type. The question becomes, do they build up quicker and faster, and how high a level do they build up rapidly? And so in most cases, the lighter textured soils, so those sands that seem to be better aerated, better drained, are the ones that soybean cyst nematode numbers will build up quicker on those in those soil types. Now, having said that, clay soils can have the same number of cyst nematode populations down the road, or same number of levels, but what may take 5-7 years on a sand may take 10 or 12 years on clays. So regardless of the soil type, cyst nematode can build up, but again, the lighter textured soils, the sands, are probably the ones you'll see it first and build up the quickest.
Okay, thanks for that. The next question I have here is, the prairie provinces have really cold winters, so will this not kill the cyst?
That's a question that we just came through a cold winter here, and we had that question asked a number of times. Again, that cyst is such a wonderful mechanism to protect those eggs, that regardless of temperatures and that, they're really hardy. They're winter hardy. I think they're probably even better adapted to the northern climates than they are in the southern U.S., where you have more biological activity, antagonist parasites during the winter, and that more soil biological activity than up here where everything is shut down with the winter and the cold temperatures. Unfortunately, we might be uncomfortable in the winter temperatures, but cyst nematode will do just fine.
Okay, that makes sense. Thanks. And just our final question for today, is biotechnology a solution now or in the future?
Oh, biotechnology. It is one of those things that, tomorrow something might be released, and we may not even know about it until it's released. And when it comes to biotechnology, we've been hearing rumors and things for 15 years or more, and there are mechanisms. There are a lot of companies and a lot of research institutions, both public and private, that are looking into biotechnology from a cyst nematode side of things, whether it is incorporating different sources of resistance or moving genes into the soybean genome that could aid in resistance or bring in some of those much needed new sources of resistance in there, or look at or incorporate maybe some type of chemical or exudate that's released in, say, a non-soybean crop such as corn or wheat, that would trigger those nematodes to hatch out of that cyst and get into that vulnerable juvenile or worm stage into the soil where they won't survive our winters in that particular case. So biotechnology, there are potential breakthroughs maybe in the future in that, but we're not there yet.
Okay, excellent. Thanks again so much, Albert. Before we end today, I'd like to once again say that this webinar has been approved for 1 CCA credit. You can submit your credits to us by emailing your first and last name and your CCA number to webinars@annexweb.com. However, if you did provide your info during registration, your credit should be processed within the next couple of weeks. And if your information wasn't provided to us, please feel free to email us and let us know. These instructions will also be found in our follow-up email. You will also be receiving a recording of the webinar within the next 24 hours. Your credit should be processed again within the next couple of weeks, and you should see our follow-up email in your inbox within the next 24 hours. Thanks again to our speaker today, Albert Tenuta, for a great presentation, and thanks again to our sponsor, Bayer, for sponsoring today's session. We hope to see you again soon. Goodbye.